Today we will start chapter 4 O-level biology 5090 and the topic is biological molecules. And first of all, in the first slide, I would like to discuss the different types of biological molecules and their chemical composition and their units. And then first of all, I would like to discuss about carbohydrates. The carbohydrates are made up of carbon, hydrogen, and oxygen. It means three elements are involved in the formation of carbohydrates, and their smallest unit is monosaccharides, or monomer is monosaccharide. And the then second type of biological molecule, proteins, which are made up of carbon, hydrogen, oxygen, and nitrogen. So these four are involved in its formation, but sometimes sulfur and phosphorus are also present in some amino acids, and their monomers are amino acids. Third type of biological molecules are lipids, and lipids are also made up of three elements. They are carbon, hydrogen, and oxygen, just like the carbohydrates. But in this case, in carbohydrates, their concentration of hydrogen and oxygen is 2 is to 1, but here their concentration is 16 is to 1 or 18 is to 1. It means in lipids, there will be the lowest concentration of oxygen. So they are different from carbohydrates. And the monomers of lipids are fatty acids and glycerol. And the fourth type of biological molecules are nucleic acids or DNA, which are also made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus or phosphate molecule. And their monomer or smallest unit is nucleotide. Now I would like to move towards the second slide. And in second slide, I would like to discuss about the carbohydrates. Different types of carbohydrates. They can be divided into two major parts. They are simple carbohydrates or they are complex carbohydrates. The simple are further divided into monosaccharides and disaccharides. Monosaccharide means the molecules consist of only one unit of carbohydrate or one glucose molecule. And in disaccharides, two molecules are joined together with the help of a bond. And in polysaccharides, many molecules are joined together in the form of a long chain. So many carbohydrate units are involved in the formation of polysaccharides. Here I would like to give you some example of these three types of carbohydrates: monosaccharides, glucose, fructose, galactose. So all they are consist of only one unit. And disaccharides, maltose, lactose, sucrose, they consist of two monomers joined together. And in polysaccharides, many monomers are joined together. And the examples are starches, cellulose fibers, and glycogen. So these are the three different types of polysaccharides which we will discuss today. Now I would going to open the next slide. And in the next slide, you can see, I will show you how the formation of disaccharides takes place from monosaccharide. So here you can see on screen, when one glucose molecule joins with the other glucose molecules, there is the formation of maltose. And the result of glucose and joining after the other glucose molecules, when maltose is formed, then a bond is formed between two glucose molecules that is called as glycosidic bond. And this glycosidic bond is formed when there is removal of one water molecule from both of these molecules. And when this water is removed, the process is called as condensation reaction. Condensation reaction. So if we hydrolyze, if we want to hydrolyze these two disaccharide molecules or polysaccharide molecules, then we have to add the water into them, and that process is called as hydrolysis. That process is called as hydrolysis, but in this case, when there is the formation of larger molecules from the smaller units, then the process is called as condensation reaction, and there will be the removal of water molecule on each addition of one molecule to the others. So when one glucose molecule joins with the other glucose molecule, there is the formation of maltose, and there will be the formation of a bond between two glucose molecules, which is called as glycosidic molecule, and this process is called as condensation reaction. Now I would like to move towards the next slide, and in next slide you can see glucose, fructose, galactose, all they are monosaccharides or monomers of carbohydrates. When in the previous slide you had seen that one glucose molecule joined with the other glucose molecule, then there will be the maltose. But when one glucose molecule joins with other fructose, or it make a bond with the fructose, again same bond, which is called as glycosidic bond. And how this bond takes place, you can see in this way this bond formation takes place. So that is glycosidic bond, and there will be the formation of sucrose molecule. And when one glucose molecule joins with the galactose molecules, then there will be the formation of lactose molecule. And all in these three cases, condensation reaction. Condensation reaction, that is the removal of water molecules. And same is the case when there is the formation of polysaccharides. Cellulose, cellulose is important part of the plants, that is the cellulose fibers make up the plant cell wall. Okay, and then the next is starch, second type of starch, which is also made up of many glucose molecules. You can see the many glucose molecules are here joined together with glycosidic bonds. So starch is stored in the plants, because when there is extra glucose in the plants, then the extra glucose is stored in the form of starch in different parts of the plant. And same is the case with the glycogen. When there is extra glucose in our body, in human body, the glucose is stored in the form of glycogen. It means all these three polysaccharides are very important. All they are joined with one another with the help of glycosidic bonds. So they have made long chains. I would like to show you in this next slide the actual their appearance. You can see these are the smaller circles. You can say these are all their molecules. Those molecules you can see. The starch is a branched structure which is made up of amylose, amylopectin. So these are the specific type of, you can say, branching system in starch. And in glycogen, further branching, also branching in the glycogen, you can say, but this branching system is more complicated than starch. But in case of cellulose, they are in the form of long chains. The long chains are also making some bonds with one another. So in this way, the cellulose make a very compact structure which give strength to the plants. So in this way, these three polysaccharides are formed from the disaccharide molecules, and disaccharides are formed from monosaccharide molecules. The second type of biological molecules are proteins. And the unit of protein, amino acids. And they are organic compounds which serve as building blocks for protein. So these are the amino acids, and amino acids make up larger protein molecules when they join together, with the help of peptide bonds. So peptide bonds will be there, and same Zeke condensation reaction will take place over here. So before going in detail, I'm going to show you structure of protein in the next slide. And here you can see a structure of amino acid. And structure of amino acid, you can see amino group and carboxylic group, a side chain, which can be any other molecule, and hydrogen molecule. So in this way, this is very simple structure of amino acid in which amino group is present, carboxylic acid group is present, COOH, and R side chain, which can be CH3 or CH2, CH3, any other side chain can be attached with this, and in this way there will be change in the structure. And then here you can see amino acids, naturally 20 amino acids are there, and these, some of them are alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, alanine. So these are some of them which are naturally occurring. So these 20 different amino acids join together to make one protein. And you can see these 20 amino acids are making so many different chains of protein. Sometimes they can be, first can be alanine, other can be arginine or asparagine, and so on. So in this way, they join together to make up one protein chain. Amino acids can make smaller, larger chains of protein. And here, next biological molecule, which is lipid. And lipid are made up of glycerol and fatty acids. One glycerol molecule will join with 3 fatty acid molecules, and there will be the formation of triglycerides. In the next slide, how do the formation takes place? When one glycerol molecule joins with the 3 fatty acid molecules, there will again, on the removal of 3 water molecules, condensation reaction. Again, condensation reaction is involved in the formation of ester bond. So here, ester bond is formed to make triglyceride molecule. To make triglyceride molecule, glycerol and fatty acids. One glycerol molecule and 3 fatty acid molecules. They will give rise formation of triglyceride, and the bond which is formed, that is called as ester bond, as a result of condensation reaction. Next, DNA. Unit of DNA is nucleotide, and you can see over here on screen nucleotide, and it is made up of a phosphate group, a pentose sugar or deoxyribose sugar, and one nitrogen base, which can be adenine, guanine, cytosine, or thymine. So one with the addition of one nitrogen base, deoxyribose sugar, and phosphate group make up one nucleotide molecule, and many nucleotide molecules join together to make one DNA molecule. And here on the screen, you can see I have shown the structure of DNA. Here you can see DNA consists of 2 strands. They are twisted together in the form of this helix. And these 2 strands, the sides, these 2 sides, which act as a backbone, They act as a backbone and they are made up of sugar and phosphate molecule. Sugar and phosphate molecule they are. And then one nitrogen base. Nitrogen base. And this nitrogen base is T. T means thymine. Thymine always make a bond with adenine and double bond is formed between them. And if there is adenine, it will always joins with thymine with double bond. It means when thymine joins with adenine or adenine joins with thymine, there will be the formation of a double bond. And this is rule of nature that thymine always joins with adenine. And in the lower, guanine always joins with cytosine and cytosine always joins with guanine. Triple bond will be formed between them. Triple bond. And this is these are the rules of nature and all they join together and they will make up a long chains of DNA. Now, the next slide, we are going to discuss about paper 4, the iodine test for starch. And for this purpose, we have to take the freshly prepared iodine and then we will take a fresh potato. We will peel off. We will take it directly as by peeling and cutting into halves or in smaller pieces, or we can crush it. And then we will add iodine solution onto it. If its color from yellowish brown, color is of iodine, or you can simply say brown color. If it is changed into blue black, it shows the presence of starch into the potato. If color is changed to blue black, if color is not changed to blue black, it remains the same. It means starch is absent. Now I would like to move towards the second type of test, which is Benedict test, which is used to find out the amount of glucose or reducing sugar in the given solution. So for this purpose, you have to take a test tube by taking 3 ml, 5 ml, any quantity of the given solution which contains glucose or not. It depends on the during the experiment. And same quantity of Benedict will be added into it, sample and Benedict solution. And then you will heat it up into a water bath. And the temperature should be above 80 degrees centigrade. So it should be above 80 degrees centigrade, and then you will heat it up. If it remains blue, it means reducing sugar absent. Reducing sugar is absent. If it is changed from blue to green, green to yellow, yellow to orange, and orange to brick red. It shows the presence of reducing sugar in the given sample. So it's very easy to find out the presence. And here I am going to show what the different colors during the Benedict solution shows or Benedict test shows. If it remains blue, no. If it is green or yellow, it means small quantity of reducing sugar is present. or the low concentration, you can say the, but in case of orange and red, high concentration of reducing sugar is present. But in the red color shows the highest concentration, and it is a confirmatory test for the presence of reducing sugar. Next is the test for protein, which is biuret test. Biuret test, first of all, we will add dilute copper sulfate solution in the sample solution, which will contain protein, or maybe protein is absent from in that solution. So it is sample solution. So we will add dilute copper sulfate solution, and then we will add sodium hydroxide solution, or sometimes we add biuret reagent, which is already mixed of these two quantities, copper sulfate solution and sodium hydroxide solutions. And then if the color is changed from blue to purple, if its color is changed from blue to purple, it means proteins are present. If the color remains the blue, it means proteins are absent. Or if it is purple, then present. So in this way, we can find out the presence of proteins in any given solution. Now I would like to move towards the last test, which is emulsion test for lipids. And here in this case, again, this in sample solution, first of all, in sample solution, we will add 95% ethanol, 95% ethanol, and then we will shake them thoroughly. And then we will add distilled water into it to make clear everything, whether there is formation of emulsions or not, or cloudy appearance is there or not. So after doing this, if lipids or fats are present in the sample, then there will be cloudy emulsion or cloudy appearance will be there. It will show the presence of lipids. Or if it remained clear solution, then absent. Now I would like to explain little more about this test in the next slide. What happened in this emulsion test? Lipids do not dissolve in water, they do not dissolve, but they will dissolve in ethanol. So first of all, we will take a sample solution and we will add ethanol, 95% ethanol. So what will happen? The lipids will dissolve into this ethanol solution, and the solution will be clear solution. Then shake the sample and ethanol so that the lipids dissolve. Pour the mixture of ethanol and sample into water. Then we will pour, after shaking, we will pour it into distilled water. Distilled water. So what will happen? Lipids are not dissolved in water. So they will make their appearance in the form of emulsion or cloudy emulsion, which will be white in color. So in this way, we are able to find out the presence of lipids in any given sample solution. So it was all about chapter 4. Please like, share, and subscribe my channel, and I will go for the next chapter in the next video. Thanks. Thanks, and Allah Hafiz.